All right, I've got 1 minute after the hour here, so I think that's a good time to start the introduction at least. I just have a few slides and then I'll be turning it over to Stephanie Henson, our speaker today. So thanks all for joining. This is another webinar for SCOR Working Group 168, the 4D BGC Working Group. We have webinars, we're trying to get to about every month. And our goal here is to enhance utility and of BGC Argo observations through 4D BGC data products. And this webinar series is a way to introduce the community to new data products and to the science being done with 4D BGC data products. So this is a photo from a recent meeting the group had in Glasgow in February. So a couple things just to introduce here. One thing is that our online data product repository is live. This is on the Biogeochemical Argo website. And we have a number of different mapped biogeochemical data products on the site. And this is an example of one of those pages from a mapped dissolved oxygen data product. This is the one from Taka Ito. So you can go on the site, you can filter by type of parameter, by area of coverage, by resolution, lots of different things, and click on a product and get all this information about them and find the data product DOI and the related publication. So we're hoping this will be a really good resource for accessing mapped biogeochemical data products from BGC Argo. The group also has a website. I encourage you to check it out. It has the schedule for our webinar series, including this one today, and some other updates about group information and group activities. And also has a link where you can join our mailing list to hear about webinars like this one and other events that the working group is putting together. This is the sign up link or sign up form for the mailing list here. And lastly, I just wanted to mention that all of our webinars are recorded and posted online. So if you miss one, you can check out our YouTube page to watch any that you missed. All right, so with that, I'll introduce our speaker today. So we had Nathan Briggs from NOC a couple of weeks ago, and now we have Steph Henson, who's going to be speaking about global patterns in organic particle loss in the mesopelagic. So this is an application of a 4D BGC data product, and I'm really excited to hear about it. So I will turn it over to Steph.
Yeah, thanks, John. Okay, let me try and share my screen. Hopefully, it was working before, so hopefully it will work smoothly again. How's that look?
That looks great.
Good. Okay. So thank you so much for having me today. And yeah, this is another edition from National Oceanography Centre. We hope to have more in the future. And for this analysis, I'm actually using Raphael's product. So I'll divert any questions about the product directly to her at the end. So we're interested in looking at small particles in this analysis. And the reason is that the majority of POC in the mesopelagic zone is actually in the small end of the particle range, but their abundance and their carbon content is quite difficult to measure using classic shipboard techniques. And so, perhaps that implies that we have a limited understanding of their role in the ocean carbon cycle. But we think that small particles could potentially be important as a supply of food to mesopelagic ecosystems, and possibly also overlooking small particles in mesopelagic carbon supply and demand budgets could make it harder to balance those budgets. So we think that small particles may well be important. We know that large particles dominate the total flux, but small particles dominate the particle abundance. And other studies have found that slow sinking or small particles could be significant contributors to flux, actually, such as this one from Chelsea Baker. And other studies have found that at least half of total POC is contained in slow settling particles. Slow settling is not the same as small, but for the purposes of this, let's just assume it is initially. And Colleen Durkin found back in 2015 that small particles were actually actively settling in the water column. They weren't just hanging out, they weren't just suspended, they were actually settling. And she concluded that they could be an important contributor to carbon flux throughout the mesopelagic. And another motivation for doing this analysis, although we can't actually address it directly with what we're going to do for a reason I'll explain later as we go along, is this long-standing discrepancy between different spatial patterns in global estimates of transfer efficiency, where transfer efficiency is the ratio between POC flux at a certain depth, say 1,000 meters or 2,000 meters, versus a much shallower depth, often 100 meters or the base of the photic zone. And what that tells us is the proportion of particles that are being transferred from the upper ocean into the deep ocean, where a high number indicates a higher transfer. So you can see in these two pictures here that there's some contrasting patterns that have been found in the literature. Some find transfer efficiency to be high at high latitude and low at low latitudes and vice versa. So for a long, long time, when people asked me what I thought was underlying this discrepancy, I put this down to the fact that shipboard observations just chronically undersample in space and time. And I think all of us who use shipboard observations are aware of that, that shipboard sampling can be very sparse and quite clumpy, by which I mean we tend to do transects, or we go back to the same locations time and time again for sustained observing systems. And because of those characteristics, we found that in this paper here, we found that clumpiness, that sparseness does result in large errors in reconstructing global flux estimates. But we also tested the ability of the BGC Argo network to reconstruct those fields. And the BGC Argo does seem to be sufficiently dense and more evenly distributed, and it does a much better job than sparse shipboard sampling. So that kind of motivated me to go back and look again at whether we could start using BGC Argo data rather than shipboard observations to answer some of the questions that have been raised. So to do this, we used Raphael's product, which is based on BGC Argo and satellite data extrapolated using a machine learning model. And it gives you a global monthly climatology on a nice grid. And this is so Raphael's product generates backscatter information, and then we convert that to a POC estimate using a depth-dependent POC to backscatter ratio. And I think Nathan talked about that on the 13th of March on his seminar, although I didn't check. So maybe, hopefully he did. Anyway, so we converted it into POC. Now that's POC concentration, and that's really important to remember all the way through this talk, that we're not talking about POC fluxes. I'm just talking about POC concentration, and that's quite important. So what do the POC profiles look like in terms of small particles? Here's just some examples, just pulled out from 3 of the pixels in 3 contrasting regions. And you can see that small particle POC, they show the expected decline with depth, as you would hope. There's some interesting differences between the areas. Again, nothing that would be unexpected. For example, POC concentration at the surface is higher in the North Atlantic than it is, North Atlantic subpolar than it is in the subtropical regions, for example. But you can also see that there's differences in the shape of the profiles and how much of the POC concentration at the surface is reflected at depth. So if we sum up over every pixel how much small organic carbon there is in those particles, we find this map on the left. So this is the POC stock from the base of the photic zone to 1,000 meters depth. And we find that when we compare these results to what Nathan's been finding, that about 90% of total organic carbon in the mesopelagic is contained in these small particles. So in the figure, you can see that there's more small particle POC stock at high latitudes. That kind of makes sense because they're more productive, and so we would expect there to be more particles at those high latitudes. So that's right. The dots indicate the months of missing data, and that's because the product incorporates satellite-derived observations, and at high latitudes in winter, then we don't get good retrievals. So the data get more sparse at those higher latitudes. Now, if we look at how small particles are being lost in the mesopelagic, a very simple metric for defining what's going on could be the ratio of the small particle concentration at 1,000 meters compared to the small particle concentration at the euphotic zone depth. So that's what we calculate here. So higher numbers mean that more small particles, as a ratio, there are more small particles at 1,000 meters than, I'm not explaining this very well. Do you understand what I mean? There are more small particles at 1,000 meters surviving from the photic zone depth than there are at low latitudes than there are at high latitudes. So this, we called it the TESP. We weren't quite sure what to call it, but it's the ratio of small particle PSC at 1,000 meters to small particle PSC at photic zone depth. And it's low at high latitudes and high in the oligotrophic gyres. So what does that mean? That means that at high latitudes, there are greater quantities of small particle POC being exported than at low latitudes, which makes sense. But a greater proportion of those small particles are potentially being lost in the mesopelagic at high latitudes than at low latitudes, which wasn't the pattern I was expecting, actually. So it's intriguing. So we can dig a bit more into it. Before we do that, I'm just going to take a slight detour into export horizons, so that the depth at which we calculate or we assume that export is happening. And I'm taking a detour because it's something that comes up quite frequently in the literature, what export horizon to use. So on the left, I've just plotted up what you've already seen, basically, which is the basically photic zone to 1,000 meters depth. And on the bottom, if I take 100 meters depth instead as the export horizon and do the same ratio, you see the difference in the bottom plot. So it is quite different. So using a different export depth has caused a large change to the patterns. Maybe it's even somewhat reversed, maybe you might say. So the differences in the low versus the high latitudes. If I use mixed layer depth instead, it doesn't make any difference. At high latitudes, if we use a fixed depth like 100 meters, it seems to overlook any remineralization that might be occurring immediately below the euphotic depth, because at high latitudes, the euphotic zone depth is typically shallower than 100 meters. Whereas at low latitudes, we see a significant production of small particles between 100 meters in euphotic zone depth. So because the euphotic zone depth is normally deeper than 100 meters. And so that's why you see this, this funny pattern. So slight detour there into export horizon. So the export horizon that you choose does matter to the results that you get. But we're going to stick with the euphotic zone depth for the rest of the talk. So in terms of thinking about that spatial pattern that you see in the top left there of the small particle ratio, think about what drives that pattern. And there's three kind of hypotheses that we could put forward. One is that the particles could be being detrained by the seasonal mixed layer pump. One is that they could be generated in situ in the mesopelagic by zooplankton fragmenting particles, or that small particles could be sinking, although slowly, or any of those. Any combination of those could also be true. We can't directly test any of these hypotheses with the data set we're using because we're just relying on the BGC Argo data, not doing anything fancy with it. So we can't directly test any of these, but it's interesting to look at some of these hypotheses and see if there's some we can rule out and some which seem more likely. So in terms of detrainment by the seasonal mixed layer pump then, this is a figure from Giorgio's paper back in 2016, which demonstrated that the shoaling and deepening of the mixed layer in the spring, which is very pronounced in high latitudes, could trap some small particles below the mixed layer, well, any particles really below the mixed layer, and then they're lost. They're just, I suppose you could call them exported, although the mechanism is different than gravitational sinking. So it's more of a sort of direct injection of particles to deeper layers. Giorgio did a nice global analysis where he found that export by the mixed layer pump is highest at the high latitudes and in spring because the seasonal changes in the mixed layer depth are much, much higher in those regions and time periods than other places. But we find in our analysis that the annual mean TESP is lower in high latitude regions. That's inconsistent with the pattern that we're seeing in this mixed layer pump driven export that you can see on the left there. However, we do find that monthly values of TESP, if we break it down into month by month, are higher during spring than summer in some regions. So the mixed layer pump could be a significant source of small particles during that winter-spring transition. I'll just show you what those months, I'm sorry, these are tiny, I apologize, but it goes from January through to December here. And hopefully you can see the changing patterns. If you focus on your favorite place, you can look through the figures and see how it's changing. Okay, so that's detrainment. The second possible driver is the in situ generation of small particles in the mesopelagic. So zooplankton activity can fragment large particles into small particles, and you can see that in this example from Nathan's paper back in 2020. Again, this is based on BGC Argo, where you see the large particles on the top and the smaller particles being generated on the bottom as a result of fragmentation. So again, we can't test that directly with this data set, but what we can do is look at whether there's any possible association between POC stock and the presence of these mesozooplankton. And so we looked at the MARIDAT database, which is a database that's been collated together where the data have been standardized and gridded to be used for model intercomparison studies and that kind of thing. So on the left, we're looking at 3 different biomes. And the blue line is the POC stock between 150 and 500 meters depth. And the red dots are the mesozooplankton biomass. And so what you can see is that there's some, similarity in the shape of the seasonal cycles, which is suggestive that generation of small particles might be occurring by fragmentation. And the seasonal peak in mesozooplankton biomass coincides more or less with the peak in small particle POC. Of course, correlation doesn't equal causation, but it's kind of, an interesting observation, if you like, that the seasonal cycle in these 2 factors are very similar, at least in the regions where we've got enough zooplankton data to make the plots. So we haven't ruled out zooplankton as being a possibility. The third hypothesis was small particles could be sinking directly. So it could be part of the gravitational flux. And this is from Colleen's paper that I mentioned already, where she used gel traps at bats at the Bermuda Atlantic Time Series site. And you can see there's size along the bottom, and then there's a particle number flux up the side. And she found that actually there was quite a large flux of material associated with small particles, and that the signal propagated through the different depths as well. So that's what's on the right-hand side, the different lines, the different colors at different depths. So she concluded that actually small particles were able to sink in this region anyway. So we can take a look at some depth differences as well to see if the signal is propagating like Colleen did. And that's what we've got here. So again, 3 different biomes, and we're going from near the surface down into the deeper waters. So from black to green to red is from surface down to deeper waters. And what we find is that generally at high latitudes and in equatorial regions, we see lags of about, well, sometimes it's 0, and sometimes it could be up to a month, and longer lags at low latitudes. But you can see that the seasonal pattern does bear a resemblance between the different depth layers. And that suggests that at least a fraction of the small particles could be sinking with speeds of around 30 meters per day if you make the calculation at high latitudes, if you assume they're just sinking straight down, and around 5 meters per day at low latitudes. And those sinking speeds are consistent with recent data compilations. And it implies that, so despite the fact that small particles are sinking more quickly at high latitudes, which generally we'd assume meant less opportunity for remineralization or fragmentation because they're sinking really quickly. That's not the spatial pattern that we actually see. We see the opposite. So we actually see that more small particles are lost at high latitudes. So that got us to thinking about why that could be happening. So if they're sinking quickly, then why are they disappearing more rapidly than at low latitudes? What we came up with was that small particle lability could be important and that it could be higher at high latitudes than at low latitudes. In other words, fresher material is being produced at high latitudes than at low latitudes. And so it's being respired more rapidly. So to think about whether this could be an option, we looked at two versions of a model because we don't have a lot of particle lability data out in the field, to be honest. So, we couldn't test this directly. But what Olivier Aumont did was to take the limited field data and develop a model where he included variable particle lability. So he took the Piskey's model, which is a well-known global biogeochemical model, and ran a version without this extra step in it, the variable lability, and then a version where he did implement the variable lability. So without on the left and with on the right. And I've calculated the same metric, this TESP ratio. And what you can see is on the next slide, I've got the observations. So the BGC Argo base is in the bottom right there. So what you can see is that without variable particle lability, the model can't reproduce the same pattern as the observations. Whereas with it, it's getting closer. Oh, there's not, there's still lots of differences, but it's getting closer to what we find in the observations. And actually between the two versions of the model runs, the spatial patterns are kind of a bit different as well. It's not just the change is quite pronounced, especially in places like the Southern Ocean, for example, the gradients as you go from the Southern Ocean up towards the equator. So that's interesting, and again, a piece of interesting evidence. But what about the role of temperature? So temperature, of course, controls metabolic rates to a large extent. And so we would expect that at high temperatures, low latitudes, that we'd have greater remineralization, just if it was just on the basis of temperature alone. So cold water's slower respiration, warm water's higher respiration, if temperature was the main thing, the only thing. So that implies that if temperature were the primary driver of mesopelagic small particle loss, then TESP should be lower in warm places. In other words, more particle loss. So we should have a negative correlation basically between the temperature and TESP. So what do we see? Oh, not what we expected again. So what we see here is there's temperature on the bottom, on the x-axis, and TESP on the y-axis, and the dots are colored by the latitude. And there's loads of dots there, but you can see that with the exception of the extreme temperatures on either end, where something different might be happening, we see a general positive correlation between temperature and TESP. So what that means is that at higher temperatures, there's actually lower particle loss, which is not what we expected on the basis of temperature being the most important driver in remineralization. So that, again, to us, another bit of circumstantial evidence that perhaps particle lability could be significant as well when it comes to shaping mesopelagic small particle loss. In fact, it looks like temperature probably isn't the main driver, because otherwise we would see that negative correlation, which we don't see, or at least it's not obvious. And so another interesting thought from a model. This is from a completely different scale of model. This is totally different. This is where these guys have taken single particles, put them in the lab, done very complicated, detailed experiments where they change the flow rate around a particle so that it's suspended. You can see these comet tails coming out, as they call them, behind the particles. And what these guys did was to measure bacterial degradation rates as a function of sinking speed in experiments. And what they found was that at faster sinking speeds, you actually get greater degradation rates, which is really odd and not at all what they were expecting, or what I would have, if you'd asked me, that is not at all what I would have expected. Because at faster sinking speeds, you assume that particles are just like flying through the mesopelagic, and there isn't really time for microbial colonization to happen and for particles to start being degraded in any meaningful way before they're just out of mesopelagic again. So this is really interesting. And even modest sinking speeds, so for that, in this experiment, modest sinking speeds, as they called them, was 8 meters a day. So it's pretty slow. So even at sinking speeds of around 8 meters per day, degradation rates were enhanced more than tenfold compared with degradation rates of non-sinking particles. Again, it's just odd because you'd expect non-sinking particles would have really fast degradation rates because it's easy for bacterial colonization to happen. But the reasoning that they give is that the flow around a particle as it's sinking, that induces removal from the particles of breakdown products, which otherwise compete for enzymatic activity. That's the idea. So that's what they found in the lab. And they implemented this into a model. And they compared this effect of this sinking enhanced degradation versus temperature in their model of single particles. And they concluded that the effect could be strong enough to overcome the effect of temperature on a degradation rate, which is really interesting. So we'll see what happens with that. They're now running some larger scale experiments rather than single particles. So it'll be very interesting to see what comes out. Okay, so all of that was kind of, as I call it on this slide, circumstantial evidence. So we don't have data sets, generally speaking. We haven't got data sets that are of sufficiently dense spatial and temporal resolution to be able to distinguish between lots of different hypotheses for what might be happening in the mesopelagic, particularly when it comes to things like fragmentation and so on. We just don't have the corresponding data sets to demonstrate cause and effect. So several mechanisms could be at play. And to distinguish between these possible drivers that I've talked about, we ideally would need data at large space and time scales on zooplankton abundance, for example. So we could potentially get that now that EVPs are being mounted on floats. We might need fragmentation rates. So we would need information about the large particle and total particle abundance needed as well as the small particle product that Raphael has produced. Maybe one for large particles is needed. If you saw Nathan's talk last week, you'll know that something is underway. It'd be really cool as well if we had sinking speeds for small particles, either through plume tracking from BGC Argo, that might be possible, or through imaging of particles. I'm not sure that we have the wherewithal to do that on individual particles at the moment, but it's something to think about for the future. And it would also be really nice to have information on particle ability. And I put question mark there because I couldn't think of how we might do that from any kind of autonomous or sustained observing platform. So if anyone has any ideas, that would be really useful. Because to be able to pick apart what's happening in this data set in terms of this loss of mesopelagic particles, small particles in the mesopelagic, ideally we would have the full suite of information, but we don't. So after all of that, do small particles matter anyway? We know that the vast majority of organic carbon flux is coming from the large particles, but that the vast majority of the organic carbon itself is held in small particles. So maybe that means that they could be important for carbon sequestration. But usually we assume that sequestration is maybe only possible for large or fast sinking particles. But if a significant fraction of small particles are persisting at 1,000 meters, especially in low latitudes, then potentially, I guess, small particle respiration could contribute to sequestration in regions of, in particularly in regions of low PAC flux. And small particles might also matter for mesopelagic ecosystems. I mean, I don't know, but I would guess that if you were living in the mesopelagic, you would be grateful for any source of food, whether it was small or large, as long as you could get hold of it and consume it in whatever way, or break it down in whatever way you preferred. Um, and especially for free living bacteria, it kind of makes sense that it might overcome the problem of a life strategy of being attached to a fast sinking particle. Sorry, particle attached bacteria might overcome the problems, of a life strategy where you attach yourself to a particle and then you sink rapidly out of your comfort zone down through the mesopelagic. Doesn't seem to me like a very good life strategy. It's sort of a one-way elevator. So, perhaps attaching yourself preferentially to small particles could be a way out of that. And for zooplankton, the huge abundance of organic carbon available in small particles could be an appealing food source, perhaps because they're reducing the amount of energy they have to expend to chase slower sinking particles. And if there's lots more of them, they'd have a higher encounter rate than for large particles. Again, all of this is speculative, but it's interesting to think about what role small particles could be playing for both carbon sequestration and for metabolism in the mesopelagic zone. Okay, so to sum up then, small particle loss in the mesopelagic seems to be greater at high latitudes than at low latitudes. And we have some circumstantial evidence for sinking and fragmentation by zooplankton could play a role in the seasonal patterns and the spatial patterns as well. And the modeling results that we use imply that particle lability could be really important. And when we compare what's happening with the patterns that we see in the temperature, maybe particle lability is actually more important than temperature-driven respiration. And we conclude that small particles aren't just a passive background of non-reactive particles that are just sort of hanging out, but they are actually being actively reworked or consumed. And we also think that perhaps small particles could be significant contributors to energy supply for mesopelagic organisms and may even contribute to carbon sequestration in low latitudes as well. So yeah, thank you for your attention, and that's it. I will try and stop sharing. I don't know what just happened there. My screen went blank. Okay, thank you.
Awesome. Thanks, Steph. That was really interesting. So we will have some time for questions if folks have any. Feel free to add any questions to the chat or to raise your hand. And I just had one, so I'll start it off to give people some time to think about their questions. So I was interested in the particle lability part, and I was curious, do you have a map, or can you describe like what the spatial distribution of the particle lability actually looked like from the model that was in that paper? I know you showed what the effect was on the test.
Yeah, yeah. So I won't be bothered trying to pull it up now, but the results are in that paper, and you can see that they found. So I should roll back a minute. So what they did was they took some observations that Phoebe Lamb had compiled, which were taken with large volume samplers, which is the best way we have at the moment, probably for sampling, like taking actual samples of small particles. And he used that as a basis for developing the variable lability model that we saw. And the pattern of particle lability was indeed, generally speaking, higher in colder regions than it was in warm regions. And as expected, lability tended to be higher at the upper mesopelagic than at the lower mesopelagic, which totally makes sense. But there was not a huge amount of data. I mean, I can't remember off the top of my head exactly how many profiles he had to work on to build the model, but It's really a handful. But that paper's 10 years old, so it might be that there's more data out there now. And it would be really
Right.
great to know if there was, because it would help immensely in trying to figure out what role particle ability might play.
Yeah, cool. Thanks. All right. Any more questions? Yeah, Guillaume, sure.
Hi, Stephanie.
Oh, hi, Guillaume.
Thanks. Super nice talk. I had a question regarding your map of the transfer efficiency. I think it was the first or the second map that you showed, and you showed the dots that were indicating the month of um sorry, yeah, the month where you had more or less data.
Yeah, exactly. Yeah.
And so I wonder if the signal that you see with the higher and the lowest transfer efficiency at higher and lower latitude could be biased seasonally because you have much fewer data at high latitude.
Yeah, so it's hard to know what might be happening when you don't have data, right? But so the general pattern, I'll see if I can share it again if it doesn't make a total mess here, um, and show you uh, the monthly figures. Do you see it okay? No, you see my notes and stuff, right? Let me find the right slide again. Okay, so if I show you the monthly figures, here we go. So hopefully
Yeah,
you're seeing
okay.
that.
Yeah,
Okay, so if you look at the monthly figures, so let's focus on the North Atlantic, say, and there are some regions where it's sort of back, it's just on the border between no data in the winter and the and what's happening in the summer. Um, and so you can see that generally, yes, the transfer efficiency is a bit higher in the winter, probably due to lags in the export of material and it, appearing at depth. So in regions where you don't have a lot of data in the winter, like at the very highest high latitudes, it could be an underestimate. But whether that's systematic enough to explain the larger scale patterns, I'm not sure, because there are regions which have coverage basically all year round, where you can still see the spatial gradients. So, for example, if you're looking from the Southern Ocean up towards the equatorial regions, the Southern Ocean itself does disappear in the middle of winter, but that gradient, you can just about see it on the edges, persists. So it's really hard to know, yeah, what might be happening when you don't have enough data to fill
yeah.
in the blanks. So.
Okay, thanks.
Other questions? All right. Well, let me just ask before we go, I guess. Um, what do you think is the number one thing over the next, call it 5 to 10 years, that we could do with BGC Argo floats that
Mm.
would make the biggest leap forward in your research? Is it more particle sensors? Is it floats in different places?
Yeah. Well, apart from maintaining the current density of BTC Argo floats, with the full suite of sensors, adding more UVP sensors to more of the floats would be great, because you've probably had talks in this series about people who've used UVP on Argo floats. And it means that you can get information about the particle size, for example, and profiles of information about the broad taxonomic groups that might be there in terms of the larger organisms. So it would allow you to get some information about mesozooplankton abundance and how particle size was changing through the water column, for example. But at the moment, there's only really a handful of floats that are equipped with UVP. And so if that could be extended more widely, that would be absolutely amazing.
Awesome. Yeah, agree. All right. Any last minute questions? Be on the lookout for emails from us for upcoming webinars. And yeah, I think we'll just thank Steph again for the great talk.
Great. Thank you so much for having
Thank you
me.
very much.
Bye, everybody.
And thanks everyone for coming. Yeah, see ya.